Hi everyone, my name's Maddie and I'm the Service Delivery Lead at EnviroDNA, and today I'm going to be discussing a new tool that we've developed called the Threatened Species Matrix. This is a framework that allows us to instantly compare, quantify and identify the most effective approach to detecting any given EPBC listed species using an eDNA approach. First, I'd like to acknowledge the traditional owners of the land that we're all meeting on and that you're watching this from. So we are based in Melbourne, which is where I'm recording this, and that is the traditional lands of the Wurundjeri Woiwurrung peoples. But the work that I'm going to talk about today and some of the case studies have been conducted all across Australia on different traditional owner countries. So a brief introduction to EnviroDNA and also to eDNA in general. Our office and lab, as I mentioned, is based in Melbourne, in Brunswick, and we specialise in, you guessed it, DNA. So since 2016, we have worked with a wide range of clients and organisations from government departments, community groups, NRMs, corporate groups, and even environmental consultants, using a range of genetic tools to detect threatened species and monitor biodiversity. For anybody watching who may not be familiar with environmental DNA, or eDNA for short, eDNA refers to the traces of DNA that are shed by all living things into their environment. So right now, as we're sitting here, you and I and everybody else are all shedding tiny little fragments of our DNA into our environments. And by capturing these fragments and extracting them from environmental samples like water, soil, even air, we can detect the organisms that are present in an environment, and that helps us better understand the suite of biodiversity in a given environment. So as I mentioned, we extract DNA from all aquatic and terrestrial environments, and by employing a range of different genetic tools and specialized tests or assays from a single sample, we can detect organisms from tiny microscopic bacteria all the way up to things as large as blue whale. There's a broad range of tools that can be used, but the two most common ones that we employ are qPCR, which is a single species presence-absence test, and metabarcoding, which is a broader approach that looks at gene regions which are shared across a group of species like freshwater fish, for example. So as a sampling method, eDNA is a really powerful tool in the tool belt that environmental practitioners, including consultants, can use when conducting assessments for biodiversity or for single species presence. One of the big challenges that practitioners face in this space, though, as we know, is that demonstrating that a particular species is or isn't present, or likely to be present at a particular site, can be really challenging. It requires a really high level of rigour and confidence, and an ability to communicate that method effectively, particularly when needing to satisfy regulators. So while eDNA is a really powerful tool, just like any sampling method, there are uncertainties that can arise. For eDNA specifically, these are things like the availability of reference genetic sequences for a species, the presence of any closely related species within the same study area, and also the need to determine the optimal sampling methods, which have to be accounted for in order to quantify the confidence of a detection or a non-detection using eDNA. So that brings us to the framework that I mentioned at the start, which is the tool we've developed called the Threatened Species Matrix. So with this matrix, we have taken all of that complexity that I mentioned and created a structured framework which can assess multiple levels of complexity for a single species, quantify that, and then provide a confidence score for the ability to detect that EPBC listed species using a particular eDNA approach. This is an overview of the steps that are involved in the matrix. So at each step, the data for that species of interest is analysed and given a score on a traffic light scale, and each of these steps have different calculations and thresholds that sit behind that. And then at the end, what we come out with is an overall score that's provided, again, on that traffic light system, to quantify our confidence in the ability of this approach to detect that species of interest. And this can be done, as I mentioned, for any EPBC listed species anywhere in Australia. So let's dive into a worked example to demonstrate how the matrix functions. Freshwater turtles are a group that we've done quite a bit of work on, particularly in collaboration with the Burnett Mary Regional Group in Queensland. There are several species that are listed on the EPBC list, including the white-throated snapping turtle and the Mary River turtle pictured here. They're quite a diverse group, and they can be really difficult to detect with traditional survey methods. So let's take a look at the white-throated snapping turtle as an example, and what we'll do is we'll walk through the steps of the matrix to determine our confidence score for detecting this species with a particular metabarcoding approach. So the first thing that the matrix looks at is genetic resources that are available. So the question is, are there publicly available genetic sequences for this species? When I say genetic sequences, what I mean is the specific base pairs of DNA that make this species unique at a particular gene region. So what we would do is plug this species name into the matrix and we would receive the following result. So for our target species, the white-throated snapping turtle, there are lots actually of genetic resources and genetic sequences available. There are 108 different sequences. So what that means is that a lot of people have collected DNA from this species and they've made those sequences available for use. So this is really good, and we would therefore get a high score for the first step here. The next part of this that we would look at is how many of those 108 sequences align with a gene region that we would actually use to detect this species. So for a vertebrate like this turtle, we would look at assays that target the regions 12S and 16S. And of those 108 sequences, we can see that 2 of them align with the 12S region and 5 align with the 16S region. So it's not terrible or great for 12S, but it's really good for 16S. The next thing that we look at is doing the exact same thing that we've just done for the target species, but for any closely related species which share the same genus, also known as congeners, and whose habitat ranges overlap. So for our target species, the white-throated snapping turtle, there are several other species with whom that shares a genus, which is the genus Elsea. But only one of those co-occur with our turtle, and that's the northern snapping turtle, Elsea dentata, which is pictured here looking very suspicious. This species also has a lot of public sequences available. There are 100 of them. And of those, if we look at how well they align with those barcode sequences, we can see that they have a really good alignment. So there's 5 that align with 12S and 8 that align with 16S. So at this point in the matrix, once those first 2 steps have both been given the green light, we're now confident that there is enough genetic information available for the target and the co-occurring congener in order for us to be able to detect both of those species. In the third step of the matrix, we dig a bit more into those barcode sequences. And what we're trying to determine here is what the most appropriate metabarcoding assay would be to use, and also how well those assays can actually tell those 2 turtles apart. So we know from the first 2 steps that both gene regions can reliably detect each of these 2 species, but the question now becomes, can we actually tell them apart, and which assay is going to be most effective at doing this and give us the highest confidence in that ability? So what we're looking for here is whether all of the sequences in each region that we're looking at, 12S versus 16S, form a nice, clean, single group that is distinct for each species. And this is where things can get a little bit messy with taxonomy, so we've illustrated this here with a phylogenetic tree. And so this is the tree here for our target species, and what we can see here is that the species sequences do form nice, clean, little distinct groups. So both the 12S and 16S vertebrate assay barcodes cleanly identify the white-throated snapping turtle. If we look at this phylogenetic tree of all of the members of the genus Elsea, you can see that all of the sequences for that species are really nice and neatly clustered in this group here in red. They're on their own branch, they're separate from everything else. So what that means is that there is little genetic variation at those barcodes within that species, because they're all nice and snug up there on the tree. The other thing that we're looking at is that there's also sufficient genetic variation between Elsea albigula, our target species, and that overlapping congener that we talked about, Elsea dentata. So you can see Elsea dentata, the sequences coming down here on the tree. And even though there is some overlap in the sequences for Elsea dentata with a few other of the members of that genus, it is still relatively clean, and it's also nice and distinct from the one that we're looking at, which is Elsea albigula. So the upshot of this, and what the matrix really condenses it down to is that we now know we have a high confidence that we can detect and also differentiate our target species, Alcea albigula, from that closely related co-occurring species using either the 12S or the 16S barcode. Once we've gotten to this point, the final step in the matrix is looking back at past detections of the species to provide some real-world confidence. So what the matrix tool does is it looks through our detection database and it identifies how many times the species has been successfully detected using those assays. And what we see for this species is that using the vertebrate 12S assay, we have detected the white-throated snapping turtle over 100 times. So that gives us really good confidence that this will actually work in the real world and that we've done it before, and we're confident that we can detect that species using that method. One of the examples of some of those past detections that I mentioned is our work with Burnett Mary Regional Group up in Queensland. So we used the vertebrate 12S assay to detect this species in the Burnett Mary region, as well as also developing a qPCR assay for the species. And we used that to help validate those metabarcoding detections for even further confidence. The qPCR assay that we developed has also been used to detect the white-throated snapping turtle in the Burnett Mary region, and that has been validated against both trapping data and expert verification. And the cool thing through this work was that a new population of the species was actually discovered in a part of the region that it hadn't been previously detected. It's a really good success story. So what we come out with at the end of the matrix is the overall result with our species, with a red, amber, green for each step and the overall score. And what you can see here is that for this turtle, it's all green, which is fantastic. This is a really positive result. So why are we using this? Why have we developed this? And how does that tool help us really address uncertainty? So quantifying the levels of complexity at each stage of the matrix and assessing that confidence helps to communicate this better to stakeholders and to regulators. And it also really clearly demonstrates the rigor behind the method employed while also simplifying that communication. Doing this at the inception of a project also streamlines the analysis approach by really quickly identifying what the best method is going to be so that you can plan and budget that project with confidence. Identifying the most robust assays for each species also leads to better results, and it can identify problems early where we may need to look at alternative tools like qPCR or other methods to get that level of confidence that we want. And then finally, the matrix provides really defensible evidence and calculations to support any submissions to regulators for targeted species surveys. So, as I mentioned at the outset of this presentation, we've collaborated with a broad range of organisations across Australia working on threatened species detection, particularly Commonwealth-listed species. So our experience working with clients in this space, seeing the challenges that they're faced with and communicating the complexities of eDNA to them is what has enabled us to provide the highest quality advice on how best to go about applying eDNA as a monitoring tool for these species. So this experience is what prompted us to develop this matrix to help quantify those complexities and provide a really clear way to communicate confidence when using eDNA for surveying EPBC-listed species, as well as tailoring the approach that we use to get the best result for a particular species. So I'd like to take you through a couple of other case studies as some examples of how we've worked with different organisations across Australia to detect different EPBC-listed species using a variety of eDNA tools. This is by no means an exhaustive list, but it's a good demonstration of the range of some of the techniques we can use for very different types of species. The first species I want to highlight is the greater glider. So these arboreal mammals occur along the east coast, and they rely on old-growth eucalypt forests with hollow-bearing trees for nesting and shelter. All of the subspecies of greater glider are listed under the EPBC Act, and the southern and central subspecies is listed as endangered. Over the years, we've occasionally picked up greater glider during vertebrate metabarcoding analyses on water samples. Moreton Bay City Council in Queensland are particularly interested in monitoring this species in their region, and we've worked with them across several projects to develop and refine a species-specific qPCR assay which provides greater detection probability and confidence, and also develop a more targeted sampling approach. So we developed the assay and we validated it against both tissue samples as well as some of those positive metabarcoding water samples as an added validation step. We've since used this assay on both water and scat samples, and we've also looked at comparing detection dog data to qPCR detection data on scat samples. And what we found was that the qPCR assay was more accurate at identifying greater glider scat than those sniffer detection dogs were. We're also exploring how to use this assay on tree hollow swabs to help identify the species this way. The next species to talk about today is a plant. This is the Border Range's lined fern. It's an epiphyte and it grows on volcanic boulders and tree trunks in subtropical rainforests. The species is critically endangered and it occurs only in a very small area on the border of Queensland and New South Wales. So through funding provided from the Saving Native Species program, Huonbrook and Wilsons Creek Landcare groups in the Tweed region of New South Wales collaborated with us and coordinated this work to develop a targeted assay which detects the species when it's in its cryptic stage, before it can be easily visually identified. So we developed this assay and we used it on swab samples which were collected by swabbing suitable microhabitat and substrates in the species range, like andesite boulders, and through this we were able to detect a potential remnant population of the species. On the same samples, we also conducted bacterial and fungal analysis using metabarcoding in order to identify any microbial associations at locations where the species was detected. And this was really important to help support site selection for translocations of the species. Up next, we've got the giant burrowing frog. So this little guy is found in Victoria and New South Wales, and it's predominantly threatened by habitat loss due to land clearing and development. The species is really unique as it burrows into the soil or leaf litter on the edge of waterways, and it lays its eggs when the burrow is inundated, which the tadpoles then escape and enter the waterway through once the burrows flood. So for this species, we worked really closely with the New South Wales Natural Resource Commission in order to support their species management plan, which aims to manage and protect priority species in areas that are impacted by native timber harvest, which includes the giant burrowing frog. So for this species, we optimised and validated a qPCR assay using tissue samples for the giant burrowing frog and for its close relatives to ensure that the assay was specific to only our target species. We also conducted occupancy modelling in order to determine the probability of detecting the species DNA in a water sample and the probability of detecting the species when it is present, because we know that replicate sampling is really important for increasing probability of detection. We also looked at sampling timing, and this is really important for employing an ecological lens to all eDNA surveys, like you would with any other monitoring method, but particularly so for this species because of its life cycle. So the sampling was conducted in 2 deployments, in both connected and disconnected pools, within the window when tadpoles would be present in the water. And what we found was that there was a high probability of detection, so over 95%, which was achieved for the species with 2 to 3 replicate samples per site, regardless of the season that they were collected in. This was really important to establish for the species in order to support that species management plan, particularly due to the low detection rates that were previously observed with traditional methods for the species. The final species I want to highlight today is the spot-tailed quoll, also known as the tiger quoll. So the mainland tiger quoll population is listed as endangered, and it's threatened particularly by habitat destruction and predation by introduced predators. The species occurs in a variety of habitats, particularly forests, and it occurs along the southeast coast of Australia. So New South Wales Parks and Wildlife have done a lot of work on this species, and we have collaborated to include eDNA assessments to support this through our sister company Caesar Australia. So for this species, we took a slightly different approach to the ones I've described so far, and we developed a genomic assay to identify individual quolls from their scats, which were collected near Cosio Kosciuszko National Park at various suspected quoll latrine sites. This approach was investigated as an alternative to traditional mark capture surveys in order to address resourcing constraints and also suspected trap avoidance behaviour by the quolls. The key benefit of this is that it's a minimally invasive measure, particularly compared to traditional trapping, and it allowed us to identify how many different quolls were actually represented through the scat samples. So we used that genomic approach, as I mentioned, and on those same scat samples, we also conducted a diet analysis using traditional metabarcoding to confirm A, how many quolls were represented by those scats, and B, what those quolls were eating. And the diet findings were broadly consistent with previous studies and also helped confirm the most common prey species for the quolls in that area. Now, of course, there's been a lot of work done to date in this space, and some of the case studies that I mentioned are examples of this, but we still have a lot more planned in order to continue learning and developing this field, including the matrix. So we do a lot of work behind the scenes on various R&D projects to help expand our offerings and also contribute to the field of eDNA and support biodiversity monitoring capabilities for various species around the world. So the key areas of focus for us as it relates to further developing the matrix are identifying priority species for new assay development. So we can use this matrix internally as well in order to identify gaps where existing metabarcoding assays may not be ideal for detecting particular species. While we also have other tools available, like the ones that we discussed in some of those case studies, this also really helps direct our R&D to identifying where new assays may be able to improve detectability of priority species of interest, and also help us identify where we may need to do some of our own sequencing to improve those reference sequence libraries for certain species. So as I mentioned, those reference libraries are constantly evolving and growing, including on public databases, but we do maintain our own reference libraries as well, and we continually improve these in order to help improve our ability to detect new and cryptic species as well. And finally, gathering more data and evidence-driven project examples will help support an evolving regulatory framework. Another avenue that we're also developing is expanding the matrix out to encompass not just EPBC-listed, but also various state-listed species, as well as priority invasive species, and then all other flora and fauna species in our database. So the key things that I want to leave you with today are that while eDNA is a really amazing tool, there is a lot of work that goes into determining the optimal strategy for using eDNA to detect a particular species in an environment. And it's not a one-size-fits-all approach that we simply apply to every single species. So our goal with the Threatened Species Matrix is to be able to quickly quantify all that complexity and provide a clear, tailored way forward with a measure of confidence that is easy to understand and communicate for any given species. So by considering multiple levels of complexity, from genetics to ecology to a spatial context, we can provide the best guidance on survey design, provide evidence to support regulatory submissions, and also streamline the process of threatened species monitoring. So if this has sparked any ideas for you, or you have species of particular interest that you would like to learn more about and maybe look at through the matrix, please reach out to us because we're always keen to collaborate with more people and develop this field. Thank you so much.